Study illustrates fasting's merits and downsides. Fasting restores intestinal stem cells but raises mice's cancer risk, according to MIT. August 21, 2024, and Trafton, MIT News. People and animals can live longer and avoid age-related diseases with low-calorie and intermittent fasting. This phenomenon has many complex reasons. MIT research shows that fasting regenerates intestinal stem cells, repairing damage and inflammation. After a fast, mice regenerate with refeeding, according to MIT researchers. In mice, malignant mutations during regeneration enhance the incidence of early intestinal tumors. Having more stem cell activity is good for regeneration, but too much can have negative effects, says research senior author Omer Yilmaz, MIT Biology Associate Professor and Koch Institute for Integrative Cancer Research member. Yilmaz wants more research to understand if fasting affects humans similarly. We still have a lot to learn, but fasting or refeeding during mutagen exposure can dramatically impact cancer risk in these well-defined mice models. MIT postdocs Shinya Amada and Sala Kowald led the Nature study released today. Promoting renewal, Yilmaz's lab has investigated gut health and fasting for years. In 2018, his team found intestinal stem cells fast on lipids, not carbohydrates. Fasting boosts stem cell growth. When and how can fasting increase regeneration? Since that paper, Yilmaz says, we've focused on how fasting drives regeneration. Is it fasting or eating after the fast that regenerates? Their investigation found that fasting reduces stem cell regeneration and refeeding increases it. One mouse group fasted for 24 hours, another fasted and then ate whatever they wanted during a 24-hour refeeding period, and a control group ate everything they wanted. After 24 hours of refeeding, intestinal stem cells expanded greatest. They outgrew non-fasted mouse intestinal stem cells. Imada says fasting and refeeding are separate states. In low-nutrient situations, fast cells use lipids and fatty acids. Refeeding boosts regeneration after fasting. Stem and progenitor cells regenerate intestinal lining with nutrients. Further studies demonstrated that these cells activate mTOR, a cell growth and metabolism signaling pathway. mTOR promotes cell protein output by converting messenger RNA into protein. We need this protein for stem cell growth. The researchers observed that stem cell mTOR activation created numerous cell-dividing polyamines. Reft cells grow. To become intestinal lining cells, Kowald says stem cells need more protein. Something great overused. Research shows highly regenerative stem cells are cancer-prone. Because they repair the colon lining every 510 days, intestinal stem cells divide fastest. As they divide, these stem cells create most intestinal precancerous cells. This study demonstrated that mice with a cancer-causing gene after refeeding had more precancerous polyps than those fasting. Cancer-linked mutations induced more polyps in refeeding mice than non-fasting mice. Note that well-defined cancer mutations were employed in mice. Yilmaz foresees human sophistication. Fasting is excellent, but eating charred steak afterward may increase your risk of malignant tumors. Fasting may benefit radiation patients with gut lining damage or other digestive issues, said Yilmaz. Non-fasting polyamine supplements for regeneration are tested in his lab. This fascinating study sheds light on the intricate relationship between food consumption, stem cell biology, and cancer risk, says UCSF and Cedars-Sinai Medical Center non-study professor of medicine, Ofer Klein. Their findings show that diet-based regeneration treatment may reduce cancer risk and examine polyamines as gut healers.